Hello students, hope you all are fine. Welcome to another lecture of microbial genetics. So you are listening to your very favorite Dr. Sandeepan Sengupta, who is an assistant professor in microbiology. Corresponding to your core paper of microbial genetics, in our previous classes and lectures, we have been discussing all about mutations, different subtypes of mutations occurring at the gene expression, that is DNA level, and at the protein translation level. We have studied about spontaneous and induced mutations, about the molecular mechanisms which work to generate, about the most prevalent molecular mechanisms which are involved in generating spontaneous mutations like tautomerism, frame shifts, AP sites generating apurinic or apyrimidine, that is lesions in DNA. Then we have studied about, next to that we have studied about a variety of mutagens, that is we have studied about chemical mutagens well known to induce mutations like base analogues and alkylating agents. We have studied about physical mutagens, which are the different classes of radiations all involved in generating mutations. We have studied about biological mutagens, or the moving genetic elements, which are also known as transposons and are the wonders, considered as wonders of microbial genetics. Next to that, we have, as I remember, I concluded my previous lecture discussing all about the DNA repair systems. So as a part of our study next to mutations, we have started discussing about the DNA repair systems. That is the repair of the damage that is generated as a result of errors in replication in course of the process of mutation. So my today's topic of discussion is about another repair system, that is about a DNA repair system, which is methyl-directed mismatch repair. Now, that is the very first repair system about which we have discussed was photoreactivation repair, and that is, as you all know, you all must be knowing now, that it is a form of direct repair which directly involves the work of an enzyme and comes into action to repair or to restore the thymine monomers, but without involving the work of any new DNA synthesis, any accession of base or accession of nucleotide as such, and thereby it's known as direct repair. Now, after listening to this talk, you'll be able to know, you'll come across the different forms of DNA repair, and you'll be able to, that is, answer to very important questions which are related to methyl-directed mismatch repair system. And of course, we'll come across various stages of the methyl-directed mismatch repair. It's a complicated process and is accomplished by the cell in 3 different stages. But before that, before we start discussing about the methyl-directed mismatch repair, let us recall a bit about the finality of the process of DNA replication. What is the need of these repair systems in a cell? Why does a cell need, or that is, why does a cell need a repair system? Or what is the need of the repair systems in a cell? The need of repair systems is to maintain the accuracy, so-called the finality of DNA replication. The process of DNA replication is very accurate in itself, in the sense that an error is introduced for about 10 to the power 9 or 10 to the power 10 nucleotides added. So what is the frequency of error in the process of DNA replication? An error is introduced, an error in replication which leads to mutation occurs in a frequency of about 1 in among 10 to the power 9 to 10 to the power 10 nucleotides added. And what are the systems which work to maintain the finality of the process of DNA replication? The basic systems are the activities of the enzyme, so-called polymerization, and 3' to 5' exonuclease activity, or the proofreading activity of the enzyme polymerase, which does the work of DNA synthesis. So the enzyme has a very specific, that is, geometry corresponding to its active site. The active site of the enzyme has a very sensitive geometry, and only a base with a correct form of geometry fits at the active site of the enzyme, after which the polymerized polymerization activity of the enzyme does the work of DNA synthesis. Now, next to the polymerization activity, another intrinsic activity that is that maintains the fidelity of, that works to maintain the fidelity of DNA replication is the 3' to 5' exonuclease activity. It is also called proofreading activity, and as you all know that this activity double checks every nucleotide added, and once a wrong nucleotide is inserted or a nucleotide takes on a rare tautomeric form, what happens is that the enzyme brings about a kinetic pause in the process of DNA synthesis, and thereby during this period of pause removes the nucleotide as a result of its 3' to 5' exonuclease activity, which is also called proofreading activity. But together, the polymerization and the proofreading activity of the enzyme do not account for the entire process of the fidelity of DNA replication. The process is accurate, has been found to be much more accurate. So it's quite understood that there must be some additional mechanisms which are generating or maintaining, which account for the fidelity of the process of DNA replication. And what are these additional mechanisms? These are actually the DNA repair mechanisms which go beyond and which work beyond and beside the process of the polymerization and the proofreading activities of polymerase, and serve to maintain the fidelity of the process of the accuracy of the process of DNA replication. That is, they work to repair the damaged bases or the mismatched base pairs, and thereby account for the overall fidelity of the process of DNA replication. And what is the overall fidelity or the rate of accuracy? The rate is that a wrong nucleotide is being added for every 10 to the power 9 to 10 to the power 10 that is nucleotides added. Once that is a wrong base is inserted for every 10 to the power 9 to 10 to the power 10 nucleotides added. Now, in our previous lecture we have studied about photoreactivation repair. Photoreactivation repair, DNA repair systems which all together work to maintain the additional fidelity of DNA replication based on the mechanisms can be grouped into two classes. One class involves direct repair, and other class involves removal of DNA, removal of mismatched bases, accession of a base, accession of a nucleotide altogether, the new synthesis of DNA. So the class which does not involve all these is the class called the class of direct repair systems, which are simply the enzyme systems that directly repair the wrongly inserted nucleotides. Like thymine dimers are repaired by a process of direct repair called photoreactivation repair. Then O6-methyl guanine is restored by a DNA methyl transferase enzyme back to normal form of guanine. 1-methylated bases like 1-methyl adenine and 3-methyl cytosine are further repaired by an enzyme system called ALKB, which is a member of alpha-ketoglutarate Fe2+ dependent dioxygenase superfamily of enzymes, and also directly repairs the bases. Now, besides these systems of direct repair, there are a variety of other repair processes, and all other repair processes would go for synthesis of new DNA, removal of mismatched bases in the form of accession of a base or accession of a nucleotide as such. One such repair system which involves all these is repair, which does a repair of mismatched bases and is called methyl-directed mismatch repair. So the mismatches or the rare mismatches left after replication are in turn repaired by a repair system called methyl-directed mismatch repair, which works or serves to repair the mismatches left after replication, adding that is increasing the frequency or fidelity of the process of DNA replication 10 to the power 2 to 10 to the power 3-fold. That is making the process of replication accurate 100 to 1000 times more and adding to its additional accuracy of DNA replication. So the repair system is methyl-directed mismatch repair, that is, which removes rare mismatches left after the process, immediately after the process of DNA replication. So the methyl-directed mismatch repair is actually meant a repair of mismatches immediately after the process of replication, and has three different steps, or works in three different stages. The first stage for the process is methylation of the parent DNA of hybrid molecule. The first step is methylation. The second step, in the second stage of the process, the variety of proteins come into action called MUT series of proteins like MUTL, MUTS and MUTH. And the third step, or the final step in the process, is the repair of the mismatch bases. Now the first stage is actually the methylation of the parent or the template strand of the daughter of the hybrid DNA, that is immediately after replication. After replication, the template or the parent strand is methylated, but the daughter strand is not. As a result of which, DNA becomes hemimethylated immediately after replication. And that is methylation of the template or the parent strand actually directs the further process of methyl mismatch repair, and actually carries genetic information which directs the process and governs the entire process. So this methylation of exclusively the parent or template strand of the DNA hybrid DNA molecule, consisting of one strand of parent and one strand of daughter, is being brought by an enzyme called DAM methylase. The enzyme called DAM methylase serves to do the work of methylation at the first stage of the mismatch repair, and in course it adds a methyl group to the N6 position of the adenines in all the 5' GATC sequences located within the ORC region of the template strand or the parent strand. So this is the first stage of mismatch repair, which generates hemimethylated DNA. Immediately after replication, the parent DNA is methylated by an enzyme called DAM methylase, which introduces methyl groups at the N6 position of adenines within the 5' GATC sequences present at the ORC region of the template of the parent DNA. So for few seconds, minutes after replication, after the activity of DAM methylase, the template strand is methylated and the daughter strand is not. So that is what is called hemimethylated DNA, and the information that is being carried by the methylated GATC sequences in the ORC region of the template or the parent DNA direct the further two stages of methyl-directed mismatch repair. Now, let us study about the second stage of methyl-directed mismatch repair. So, immediately after the process of replication, the first stage of methyl-directed mismatch repair involves methylation of the template strand, and the GATC sequences are specifically methylated. Now, the mismatches which are within the vicinity of the 5' GATC sequences of the template of the parent strand, that is lying about 1000 base pair away, are efficiently, that is even at a distance of 1000 base pairs, can be efficiently repaired as a result of the process called this mismatch repair. Now let us move on to the second stage of this process of mismatch repair. In the second stage of this process of mismatch repair, three proteins come into action which are known as MUT series of proteins meant for mismatch repair, and to name them, they are MUTL, MUTS and MUTH. Now MUTL and MUTS proteins together form a complex, and this binary complex binds to the methylated, that is 5' GATC sequences within the template or the parent strand. So as a result of the activity of MUTL and MUTS, that is the DNA on both the sides of the methylated sequences is threaded and looped following replication. So MUTL, a complex of MUTL and MUTS proteins, binds to the methylated 5' GATC sequences present in the template or parent strand. Now MUTH, another protein which has an endonucleolytic activity or an endonuclease activity, binds to MUTL. MUTH protein binds as the third protein which comes into action. It binds to NUTL and has an endonuclease activity which is inactive, or which till it encounters a methylated 5' GATC sequence in the template or the parent strand. So NUTH has a site-specific, that is, has an endonuclease activity, but NUTH, the endonuclease activity of NUTH comes into action only when it encounters a hemimethylated DNA, that is, of hemimethylated DNA in the template or the parent strand. So on encountering methylated 5' GATC sequences in the parent or the template strand, NUTH introduces a cleavage in the but not in the template or parent strand, but in the daughter strand. So this is the endonuclease activity of the NUTH, and with this cleavage introduced by the endonuclease activity of NUTH protein in the daughter strand ends the second stage of the process of repair. So according to the direction of the mismatched bases, that is, with respect to the cleaved site that has been cleaved by NUTH, in the third or the final step of methyl-directed mismatch repair, the mismatches are repaired. So that is, mismatches are repaired depending upon whether they are located on the 3' side of the cleaved site in the daughter strand, or they are located in the 5' side of the cleaved site in the parent strand. So we'll continue our discussion with methyl-directed mismatch repair. I am going for a very short break, and in this part of my lecture I have a very interesting activity for you all. Please search about the proteins corresponding, which are homologous to NUT series of, that is, NUTL and NUTH and NUTS homologs are the proteins which do exist in eukaryotes, like the NUT proteins exist in bacteria and perform similar functions in yeast, humans and other eukaryotes. So keep on doing this activity. I am returning back after a very short break. So see you all on that side of the break.